Okay, let's talk about the cells that make up the nervous system, which are called neurons and glia. But before we do that, let's talk about a little bit more history. So, the discovery that the brain and the spinal cord and the rest of the nervous system were made of specialized cells goes back to, of course, when the microscope was invented and people started looking at all the different parts of the body under the microscope. And but then later, several stains were developed that let you look at tissues in the body and give them a color that lets certain cells stand out. So if you just take a piece of pretty much any part of the body, could be any organ, and slice it really thin, you can put that slice on a microscope and you can look through the microscope at that slice. But most tissues of the body, most cells, are actually transparent if you slice them really thin like that. And so in order to actually see any cells or any microscopic structure, you have to put some sort of stain. So there are lots of different stains you can use to look at cells. They can be, mostly they're just pigments that you wash the slide or the tissue in, and they get embedded in the cells or in the tissue. And different stains label different things. Just depends on the chemical property of the stain and on the thing you're trying to look at. So some stains stick to the cell membrane, some stains stick to proteins. There's lots of different examples. One important example for the brain is this stuff called the Nissl stain. Sometimes it's called crescel violet because it's this purple pigment, and it has the ability to basically stick to nucleic acids. And so, of course, most of the nucleic acids in a cell are in the nucleus, but there are also nucleic acids in the cell body that are part of ribosomes mostly. And so it was developed by this guy named Franz Nissl. And in the brain, the Nissl stain is really useful because, again, it stains nucleic acids, which means it stains the nucleus, so it stains all the cells. But it turns out that the two types of cells in the brain, the neurons and glia, show different patterns of Nissl stain. So the neurons, when they're labeled with Nissl stain, the whole cell body is labeled. You can see lots of purple, and so all these are all neurons. So you can see the whole cell body, and the whole thing is labeled. You can still see the nucleus, so the nucleus is still visible and distinguishable, usually depending on the how good the stain is. Here's another example. You can see again. Here's the cell body, and here's the nucleus. Meanwhile, glia, only really the nucleus gets labeled. So these smaller little structures are the nuclei of glial cells. Now, the reason for this we'll get to later. Mostly it's because neurons just have a lot more ribosomes, especially ribosomes attached to the endoplasmic reticulum. But what's really useful about this is that it lets you, again, distinguish the neurons from the glia, so you can tell that there are two types of cells. And again, this was kind of a discovery made using the Nissl stain. And it also lets you visualize what we call the cytoarchitecture of the brain. So cytoarchitecture just means the number and type and arrangement of neurons in different parts of the brain. In fact, the Nissl stain shows you that different brain regions have different cytoarchitecture, and that difference in cytoarchitecture actually reflects differences in function. In fact, in this image here, you can even see that this region here in this slice, the neurons are kind of spread out more or less, with lots of sort of extracellular space between the neurons. And then there's this layer that sort of surrounds it, where you have neurons that are much more densely packed. So this really dark region, these are all neurons that are just packed really tightly together. This is actually part of the hippocampus. So again, this Nissl stain lets you visualize that. Another really important staining technique is called the Golgi stain. It was discovered by this guy Camille Golgi, and the stain basically involves putting a piece of brain tissue into a solution containing this special silver salt. And what happens is silver granules kind of precipitate onto or into the cell body and or into neurons. And what happens is, for complicated reasons, not every neuron gets labeled, only a certain random percentage of them. And that's important because if you actually stained all of the cells with the Golgi stain, you can kind of see here it's this dark brown, almost black pigment here, then pretty much the entire piece of tissue would be black, because even though you're seeing a lot of empty space there, that empty space is filled with neurons, you just can't see them. But because this Golgi stain just randomly labels some cells and not others, it makes it so that you can see those cells. And importantly, it actually labels the entire cell. So not just the cell body, but the neurites that come out of them. So a neurite is just a projection that comes out of the cell body. And Golgi and others that use this stain identified two types of neurites. So all neurons have this long, thin process called an axon. And so this is the axon of this cell. Of course, you can only see part of it because, again, this is a slice. So you're, it's only visible through this part of the tissue that is sliced here, but it would in reality be a lot longer than that. And then these processes that come out from the cell body that branch and sometimes have these little spines on them that are called dendrites. And those processes, if you trace them, which Golgi and others did, then you can see that they go all over the nervous system and they connect to other cells. So if you were to follow this axon long enough, eventually it would come in contact with another cell, either at the cell body or at a dendrite. And so it became pretty clear pretty much immediately that this is how neurons in the brain and the rest of the nervous system communicate with each other. Signals traveling from one cell to another along these neurites. So in fact, Golgi proposed that these neurons, or these neurites, are continuous with each other. So that means that if you trace this axon, it never really ends. What it does is just fuse with a dendrite of another cell, and they're all in this continuous network. And this was called the reticular theory. Reticular basically means network-like. And then that's what the brain consisted of, just this vast network of these cells connected together, kind of like the circulatory system. So if you know how the circulatory system works, your blood vessels are all continuous with each other. Blood flows from one part of the body to another directly. The only problem with this theory is that it would be an exception to what we call the cell theory, which is that all the tissues of the body are made of cells that are all physically discrete from each other. And so, but that's what Golgi proposed, just based on the images he saw. But then another, these actually, Golgi and Ramon, and this guy, Santiago Ramon y Cajal, were both Spanish neuroscientists. And Ramon y Cajal, his last name is Ramon y Cajal, sometimes he's just called Cajal, but he looked at pieces of brain tissue, lots and lots and lots of images or slices of brain tissue using the Golgi stain. But what he did is he looked at, again, probably thousands of these images, and he took made very careful drawings of them. So this was, of course, long before cameras were invented, or at least before you could attach a camera to a microscope. So he just looked through the microscope, and then with a piece of paper and a pen next to him, just drew what he saw. And he again did thousands and thousands of these drawings, extremely detailed, and identified lots and lots of different types of neurons based on their shape and based on the branching of their dendrites. And he proposed that neurons were not continuous with each other, that instead the ends of axons terminated very, very close to the dendrites or the cell body of other neurons and came in contact with them, but didn't actually fuse together. And so neurons, he proposed, communicated at these contact points, which we now call synapses. but that they weren't actually fused. So neurons were not an exception to the idea of cell theory. And he called this the Neuron Doctrine. Of course, now we know that the Neuron Doctrine is, in fact, neurons do adhere to cell theory and that they are discrete. But Golgi and Cajal actually, kind of ironically, shared the Nobel Prize for their discoveries. But the two of them disagreed, again, about this question of whether neurons were continuous or communicated by contact. And in fact, probably didn't really like each other very much, but that's just how science goes sometimes. So the neuron is what we will talk about next time.